Good morning, everyone. Thank you for joining us today for the third installment of our webinar series demonstrating the utility of 3D cell culture, with today's presentation focusing on EpiIntestinal, MatTek's model of the human small intestine and its impacts on the preclinical testing pipeline in multiple industries. We will have a Q&A session after the presentation, so please feel free to send us your questions. And this webinar will be recorded, and you will receive a link to access it after its conclusion. And we'll also be sending out a survey for feedback and to give you a chance to let us know what you'd like to see from MatTek in the future. It's my pleasure to introduce our presenter to you today. Katie Dunnick has her PhD in pharmaceutical and pharmacological sciences from West Virginia University, where she researched nanoparticle toxicity in pulmonary models. And following her PhD, she spent time researching genotoxicity and developed a unique assay to measure double-stranded breaks. She then specialized in primary cells isolation prior to joining MatTek. And I'll now hand it over to Katie to begin the presentation.
Thank you, Margaux, and thank you everyone for attending. Before we get into the content, I thought it would be helpful to introduce MatTek as a company. So MatTek was founded in 1985 and began producing human tissue models in the early 1990s as a replacement for animal models. Since then, we've become the leaders in tissue model creation, and we now offer 15 models with more in R&D. We've also expanded our portfolio to include cells and media, as well as cultureware products to provide researchers with custom solutions to their complex research projects. In 2021, we were acquired by Babineau, the largest bioconvergence company, and we now have the resources of multiple sister companies to solve our customers' challenges. So MatTek 3D models provide an alternative for animal testing. Before I discuss MatTek models in detail, I'll highlight some of the current challenges with these animal tests. MatTek models provide an alternative for animals and have high in vivo relevance. This is important because more and more data has come out that demonstrates animal models do not translate well to humans due to differences in genetic, molecular, and cellular responses. Additionally, animal models are expensive and time-intensive, unlike MatTek 3D models. The lack of translation between animals and humans has gradually shifted regulatory and policy standards, such as the 3 policy changes listed. This includes the European Union's ban on animal testing for cosmetics and the FDA Modernization Act, which was introduced to the Senate in 2021. And would allow for use of human-relevant testing for drug development processes. These changes are vital as the current drug development process is not optimized and still uses animal models to predict human responses. And currently, about 90% of the drugs that enter the drug development pipeline fail prior to market approval. Due to the high rate of attrition, it currently takes 12 to 15 years and about $2 billion to develop a new medicine. With improved methods and alternatives to animals, the goal is to optimize this process and improve translation between early results and human responses. The main challenge in the drug development process is the translation gap, referred to in the pharma industry as the valley of death. This is where most drug candidates fail. And with the accumulation of drug failure here also comes the accumulation of significant lost time and lost capital investment. The valley of death spans the preclinical and early clinical phases of drug development. Preclinical testing is essential to address several very important questions about a new drug. Questions such as, what is the drug doing in the body? And what is the body doing to the drug? And what is the safety profile of the drug? Many preclinical tests are currently conducted using animal models, while alternative models, such as those provided by MatTek, provide a more human-relevant option for preclinical efficacy and safety assessment to answer the questions I just mentioned. The failure seen during the drug development process indicates the need for improved preclinical tests. MatTek has therefore focused efforts on developing 3D models that utilize human cells to provide human-relevant tissue models as an alternative for animals. As regulatory changes continue to shift away from animals as a requirement, the challenges currently seen with animal models and poor translation to humans will result in vitro models that can determine human-relevant responses in a cost-effective and a humane manner. So now that I've described the current challenges in the drug development process and the future of preclinical testing to use human relevant 3D models, I'll go into how MatTek tissue models are made. To create our 3D models, organ tissue is received and the cells are isolated. We then expand those cells to create a large bank of the same donor. This allows us to continue using that same donor over years for consistent model production. After the expansion of those cells, we cryopreserve the cells, and then we can thaw them when we're ready to create the tissue. The cells are seeded onto cell culture inserts and allowed to differentiate and stratify over time in culture. We then provide customers with a fully differentiated human tissue model that's ready to use upon arrival. As I mentioned, MatTek currently offers 15 human tissue models with more in development to meet the growing field and assist our customers in replacing animal models with human relevant 3D tissues. We offer models to recapitulate the oral cavity, ocular surfaces, pulmonary organs, skin, including both healthy and diseased, and intestinal and vaginal organs. Today's webinar will focus on our EpiIntestinal model and how this has been used to assess pharmaceutical safety. The models are created in various size cell culture inserts depending on the model and specific research endpoints. Our epi-airway, epiderm, and EpiIntestinal models can be grown in 96-well plates to allow for high throughput screening. These 3D models can be shipped globally, either from our site in Massachusetts or our site in Slovakia, and are shipped on a weekly basis for timely delivery. In addition, 3D models provide many advantages over animal models and 2D models. The models are all grown from primary human cells, which ensures high in vivo relevance, as demonstrated by the in vivo structure and function that's measured and that I'll show you today. The models are highly reproducible, which is allowed for regulatory acceptance for various toxicology endpoints and animal model replacements. The models are easy to use, and because they're all grown at the air-liquid interface, topical and basolateral exposure is possible, which allows for more in vivo relevant exposure methods. Compared to animal studies, these models are also cost-effective. Now that I've given an overview of the model generation, I'll go into greater detail regarding the EpiIntestinal model, then compare EpiIntestinal to the standardly used CACO2 cells, and then detail a specific study that utilized EpiIntestinal for pharmaceutical safety assessment. MatTek's EpiIntestinal model is a 3D model of the small intestine that displays epithelial polarity and is one of our latest innovations that will help in redefining preclinical testing. Our EpiIntestinal model is heavily utilized in GI toxicology testing, drug metabolism and delivery studies, inflammation and fibrosis assessments, as well as wound repair applications. The model forms villus structures and includes functional tuft cells, goblet cells, and Paneth cells. The stem cells that are present allow for the model to continuously self-renew and allows for the model to be cultured for up to 6 weeks. The standard model is grown from cells from the ileum. However, if you're interested in the jejunum or duodenum, we do offer these cells as an option. The EpiIntestinal model is offered as both a partial thickness model, which contains only the epithelial cells, or the full thickness model, which contains epithelial cells and fibroblasts. Both models form in vivo-like villi structures, and both models can be purchased in either 9mm inserts or 96-well plates, depending on the endpoint of interest. The EpiIntestinal model has been well characterized within MatTek, as well as by some of our customers, to further demonstrate the in vivo relevance. For today's webinar, I'll share a subset of this data. If you're interested in learning more, please reach out and we'd be happy to share that data with you. As I mentioned, the EpiIntestinal model contains functional small intestine epithelial cells that have appropriate polarity, are cytokine 19 positive, form brush borders, and form nice tight junctions. The fibroblasts maintain position beneath the epithelial cells and express vimentin in our full thickness model. The model contains tuft cells, which are the chemosensory cells, mucus producing goblet cells, secretory Paneth cells, and LGR5 positive stem cells that allow for the model to continually regenerate. qPCR data that we collected at MatTek demonstrates that the EpiIntestinal partial and full thickness models, sometimes referred to as SMI, express various phase 1 enzymes at physiologically relevant levels, making this model an ideal in vitro system to model drug delivery and metabolism. The qPCR data on this slide compares the small intestine explant tissue with the 2 different EpiIntestinal models for a wide range of common enzymes found in the small intestine. Further, PCR analysis of various drug efflux transporters, including P-glycoprotein, BCRP, MRP1 and 2, demonstrates high levels of these transporters in the EpiIntestinal model as well. So the functional cells, efflux transporters, and drug metabolizing enzymes makes EpiIntestinal an ideal in vitro alternative for current animal models. However, current alternatives for the small intestine generally rely on various cell lines rather than primary human cells. The most commonly used alternative are Keiko2s. So for the next few slides, I'll focus on how EpiIntestinal compares to Keiko2s to highlight why a 3D primary human model provides an ideal alternative that more closely recapitulates the human small intestine. As I detailed in the past few slides, the EpiIntestinal model is made from primary human small intestine cells that have high metabolic enzyme expression similar to in vivo tissue. The epithelial cells form physiologically relevant tight junctions, which allows for modeling of both highly and poorly absorbed drugs. The model forms the crypt-villus structure and includes mucus-producing goblet cells. At MatTek, a single donor is used to make the model over years to allow for lot-to-lot consistency. On the other hand, Keiko2 cells are best for modeling highly absorbed drugs, while poorly absorbed drugs are not well modeled due to the tight junctions in Keiko2 cells being non-physiologically relevant. Since it's estimated that about 60% of marketed drugs are orally administered and 90% of those are absorbed in the small intestine, it's important that an in vitro-based model be predictive for both poorly and highly absorbed drugs. Keiko2 cells also weakly express metabolic enzymes and are not well suited for assessment of first-pass metabolism. The Keiko2 model is cultured in 2D and lacks the crypt-villus axis that's important in fluid and ion transport, and because they're a cell line, they do not have mucus-producing cells. As with many cell lines, continued use and clone selection has resulted in interlab differences in the Keiko2 results. So as I mentioned, the metabolic enzymes found in the EpiIntestinal model versus Keiko2s are quite different. In this study, researchers assessed first-pass availability with 2 model drugs, midazolam and estemazole, and they were studying drug metabolizing enzymes in both of these models. The data shown on this slide demonstrates apical to basolateral transport of midazolam or estemazole in either the EpiIntestinal or Keiko2 cells. Cells exposed to midazolam were assessed for the presence of midazolam and 1-hydroxymidazolam, which is the metabolized version of midazolam. And this study was conducted both with or without the selective CYP3A inhibitor CYP3azide, which is shown in the top 4 graphs. Midazolam availability was decreased in the EpiIntestinal model compared to the Keiko2 cells and was increased in the EpiIntestinal model following CYP3azide treatment. Additionally, a substantial amount of 1-hydroxymidazolam, the CYP3A4 selective metabolite, was detected in EpiIntestinal and suppressed with the inhibitor compared to Keiko2 cells. This data further demonstrates that the low levels of CYP3A4s in Keiko2s. And then the second part of this study, they assessed estemazole apical to basolateral transport in the presence or absence of CYP2J2 competitive inhibitor Avastin. Both EpiIntestinal and Keiko2 models showed similar availability of estemazole, and availability was decreased in the presence of the Avastin CYP2J2 inhibitor. Following the results on the previous slide that assessed the drug metabolizing enzymes CYP3A4 and CYP2J2, activities of additional drug metabolizing enzymes were conducted. In this portion of the study, the researchers exposed EpiIntestinal and CACO2 cells to various substrates at different concentrations. The substrate concentration that approached enzyme saturation was used to compare the drug metabolizing enzyme activities between the two models. The most prominent difference the researchers found between the two models was CYP3A4 activities, which is demonstrated by exposure to midazolam and testosterone. CYP3A4 accounts for about 80% of total CYP content in the human small intestine, which makes this decrease found in the CACO2 cells even more impactful in terms of in vivo relevance. All of their CYPs were either increased or similar in EpiIntestinal compared to CACO2s, with the exception of CYP1A2, which is very minimally expressed in human intestine, as well as the EpiIntestinal model, and was 7 times higher in the CACO2 model. This indicates that there's high in vivo relevance of the EpiIntestinal model compared to CACO2s. The intracellular accumulation of metabolites was also measured in the cell lysates and demonstrated very low levels that were comparable between the two models. Enzymes that are involved in phase 2 biotransformation and carboxyl esterases were also assessed comparing EpiIntestinal and human intestinal mucosa. In vivo, the extensive glucuronidation in the intestine is a major reason for oral availability of drugs is decreased. In this portion of the study, the researchers used raloxifene and ezetimibe to assess intestinal glucuronidation, as these drugs are known to be extensively glucuronidated in the human. Apical application of the two drugs demonstrated the presence of glucuronides in the EpiIntestinal model and indicates that it has functional glucuronidase. Both the human intestinal mucosa and EpiIntestinal model only demonstrated glucuronidation of ezetimibe, while both models resulted in glucuronidation and sulfation of raloxifene, which was exactly what the researchers expected. The researchers concluded that the metabolite pattern for both drugs was similar between the human intestinal mucosa and EpiIntestinal samples, which further demonstrated the high in vivo relevance of the EpiIntestinal model. As I mentioned, CACO2 cells are known to poorly distinguish between low and moderately absorbed drugs. This study assessed the permeability of 3 compounds with low, moderate, or high absorption in humans, comparing the rates between the EpiIntestinal model and CACO2 cells. The tissues were exposed to the compounds for 2 hours, and then samples were assessed for drug concentration in the prospective chambers. The results demonstrated that EpiIntestinal was more predictive of human absorption since CACO2 cells were unable to discriminate between the low and moderately absorbed drugs, while the permeation in EpiIntestinal scaled with human absorption values. Following that initial study on the past slide, of the 3 compounds, 11 additional compounds with known human absorption rates were tested in both of the models. The 11 compounds align with the FDA recommendations for absorption assessment. As demonstrated in the SMI or EpiIntestinal and CACO2 columns, the EpiIntestinal model correlated with the human absorption data better than CACO2's. The next slide does a better job, I think, of demonstrating this correlation. So this is the correlation data here, which I think is better represented, that demonstrates that EpiIntestinal had an R-squared value of 0.9, while the CACO2 cells had an R-squared value of 0.7 when the absorption values were compared to known percent human absorption data. This data helps further demonstrate that the in vivo relevance of the EpiIntestinal model is improved compared to CACO2 cells. So following the absorption studies, suitability of the EpiIntestinal model to study drug-drug interactions was also assessed by these researchers using substrates for intestinal efflux transporters. Permeability experiments were performed to measure both apical and basolateral to apical absorption, and an efflux ratio was calculated. Then the experimental results were compared to efflux ratios following exposure of the tissue to various efflux transporter inhibitors. In the absence of the inhibitors, the efflux ratio for the drugs was greater than 2, which indicated to the researchers the involvement of these transporters in drug transportation. After the inhibitor addition, the bioavailability of the drugs decreased, which was noted by the decreased efflux ratio. The results correlated with decreased drug bioavailability in vivo and further demonstrated the in vivo relevance of the EpiIntestinal model. So in addition to the studies that have assessed in vivo relevance of EpiIntestinal, Peters et al. At AstraZeneca, in collaboration with MatTek, conducted studies to understand the utility of EpiIntestinal to predict gastrointestinal toxicity and provided researchers with an alternative model for drug development process. So drug-induced gastrointestinal toxicity, or GITs, which includes nausea, vomiting, constipation, and diarrhea, is among the most common drug adverse events seen during clinical trials. Most often, the GITs are not life-threatening, so clinical trials focus on management of symptoms or dose reduction to avoid drug attrition. While drugs are not necessarily halted, the patient quality of life, compliance, and overall efficacy can be impacted. Therefore, it would be more ideal if GITs were addressed and caught during preclinical studies. So interestingly, the GITs are actually one of the least likely side effects to be caught during preclinical studies, potentially due to poor correlation between rats and dog preclinical results and actual human clinical results. While primate studies have improved concordance, ethical as well as cost concerns limit the potential for higher order species testing of all of the drugs, with the exception of the final drug candidate. Therefore, it's necessary that we have improved preclinical tests that more closely represent the human gastrointestinal tract to detect GITs earlier in the drug development process. The Peters et al. Paper that conducted the studies I'll discuss on the next few slides addressed the need for improved models and said that if an in vitro test is to be used and be effective for preclinical studies, it must be functional, organ-specific, and have the ability to generate kinetic data. As I mentioned, Peters et al. Detailed that a model must provide functional data, be organ-specific, and provide kinetic data for repeat dosing studies. The researchers therefore selected EpiIntestinal for their studies using TEER, or transepithelial electrical resistance, as a functional endpoint to mimic barrier function disruption as a predictive correlate for diarrhea. Given that the EpiIntestinal model can survive in culture for at least 6 weeks, it can also be used to collect kinetic data from repeat dosing studies. And it's also organ-specific, as the cells are primary human small intestine cells. So as I mentioned, rat and dog preclinical trials do not accurately correlate to human studies in terms of GITs. This data comes from Peters et al. And demonstrates this fact and shows clinical results compared to the animal model results. The results shown for 4 AZD compounds tested in either rats or dogs before moving to clinical studies. And these animal studies underpredicted the clinical diarrhea observed for those compounds, and only AZD 3409 was detected at the max tolerated dose in dogs, and AZD 8931 was detected at the dose limiting toxicity in rats. The researchers then used EpiIntestinal to test these same 4 drugs and found that the EpiIntestinal model would have accurately predicted the incidences of clinical diarrhea better than the animal models, further demonstrating the importance of relevance when predicting clinical results. In addition to the previous 4 compounds that were tested, the Peters group also used EpiIntestinal for the detection of compounds that induce gastrointestinal toxicity, specifically diarrhea in humans. The researchers exposed EpiIntestinal to 31 widely prescribed drugs known to either induce or not induce instances of diarrhea. The tissues were tested at 4 concentrations under blinded condition, and TEER and MTT values were obtained after either a 48 or 96-hour exposure. This data was used to determine a prediction outcome score, which is not shown on this slide but can be found in the publication if you're interested in learning more about that. The results from this study demonstrated that EpiIntestinal has high sensitivity specificity, and accuracy to predict compounds that induce GITs in the human. Also interesting is that the EpiIntestinal model outperformed CACO2 monocultures for predicting drug-induced GIT toxicity. So in addition to testing commonly prescribed drugs, another 4 AZD drugs were tested to obtain kinetic data solely by measuring TIR. As I mentioned, measurement of kinetic data is important for GIT detection in alternative models. So this portion of the results allowed the group to obtain 5 key data points. The inter-day consistency was demonstrated by consistent TIR readings throughout the experiment in the 0.1% DMSO treatment group that's shown in green. Drug washout was measured, and after a 3-day exposure to the drug and an apical rinse followed by monitoring of barrier improvement. The viability was monitored for 42 days and showed no significant decrease in TIR values for the vehicle control, again shown in green. Additionally, the tissues were also compatible with repeat dosing on days 3 and 21. In addition to repeat dosing, they were also able to measure the different response onset and recovery as seen with the treatment of AZD1 in yellow and AZD8931 in blue. Both of these compounds impacted the barrier function of the tissue and were slower to recover after a second treatment. The results from this study further demonstrated the potential for using an in vitro assay to assess preclinical GITs. So today I showed you a few examples of data collected by Peters et al. However, the study conducted was much more extensive than what I've shown, and I would recommend reviewing the publication to see all of the work in greater detail. We'll be sending that out after the webinar. From the studies, the authors were able to conclude that the EpiIntestinal model provides a good in vitro model to predict GITs in the human and can be used to model repeat dosing kinetics to optimize compound pharmacokinetic or clinical dosing schedules. Additionally, EpiIntestinal better predicts GITs using TIR than commonly used animal models and provides improved translational accuracy and sensitivity for marketed and non-marketed drugs. The authors also discussed how EpiIntestinal allows for both apical and basolateral exposure and how TIR can be used, which allows for repeat readings in a non-destructive manner. So the last few studies demonstrated the value of EpiIntestinal in the drug discovery process, as well as detailed characterization studies that we've conducted. So to recap, the model mimics the structure and function of the human small intestine and contains LGR5 positive stem cells, which allows for continued tissue regeneration. The model can be cultured for weeks with maintained viability, which allows for repeat dosing and kinetic modeling of both acute and chronic exposures. And finally, the high metabolic enzyme expression in active transporters makes this model ideal for use in preclinical drug discovery research. So in addition to the EpiIntestinal model, MatTek is currently developing a new model, EpiColon, that will help recapitulate the human colon. MatTek's EpiColon model is currently in beta production and is expected for release in spring of 2023. The model has structural architecture similar to human colon tissue and produces mucus on the apical surface. The model forms a polarized tissue structure with fibroblasts on the bottom and epithelial cells layered on the top. Initial data that's been collected by MatTek demonstrates that EpiColon has functional drug transporters and drug metabolizing enzymes and indicates that this model will be a promising tool for drug development and microbiome research. So initial data using EpiColon has also shown promise in testing various irritants that can mimic colitis-like diseases. These two graphs represent EpiColon exposed to various irritants, including indomethacin, DSS, and SN38. The compounds were exposed to the tissue basolaterally for either 48 or 120 hours, and then TIR was measured following the treatment and compared to the no treatment control group. Results indicated that the EpiColon model is able to respond to various irritants and may provide an ideal in vitro alternative model to study colitis-like disease and drug development. As I mentioned, this model is expected for full release in the spring of 2023, so please reach out if you have more questions and would like to learn more. So as discussed, MatTek models are being utilized in the preclinical space. In addition to this, researchers are using these models internally to conduct these studies. MatTek also offers in-house contract services where we work with our customers to build unique assays using our 30+ years of experience. In fact, a lot of the studies that I showed today that were conducted in the Peters et al. Paper were a collaboration study between MatTek and AstraZeneca, where a lot of those studies were actually run by MatTek in-house. So we thank you for your interest in our webinar today, and we look forward to answering your questions. And as Margaux mentioned, a packet will be sent out afterwards following the conclusion of the webinar.
Thanks so much, Katie. That was a great presentation. We do have a handful of questions. I think we'll be able to get to all of them. But if you have additional questions, please feel free to submit while we're going through the Q&A session. The first question, which I think was answered towards the end of the presentation with your slides on EpiColon. That question was, are large intestinal cell models available? And EpiColon, as Katie mentioned, is anticipated to launch in the spring of 2023, and we're happy to reach out to you with some more information on that model. Another question is, how long can I leave my compound on the tissues when dosing them on the apical side?
Yep, that's a great question. So that's going to depend a lot on the volume that you need to use. So you don't want to necessarily suffocate the tissues. And it's also going to depend on the toxicity level of the compound. So for some of the studies that people do, they leave them on for 24 to 48 hours. For other studies, if it's really toxic, it might be less. Physiologically relevant is what we aim for. So what you expect it to be for exposure in the human is what we would work towards in this model. And the sales team is the technical sales team is great at helping answer these questions as well if we know more about the product.
Okay, next we have a couple of questions that are sort of related regarding using the full thickness model with fibroblasts versus the partial thickness. And one question asked, when should I use the full thickness model, or when should I use the partial thickness model? And then what is the response of each for drug permeability?
Yep, so fibroblasts are a great option if you're looking for more of the immune response. This is going to be dependent on your study endpoint. And then the second half of that question was, how does it differ? Was that correct, Margaux?
Yes, well, the differences between the models specifically for drug permeability.
Okay. Yeah, so for drug permeability, it's going to slightly change. Again, the technical sales team is really helpful in determining what's going to be best, but we do have internal protocols on how to use these models for those types of studies.
Okay, next, is it possible to analyze inflammatory cytokines released from the tissues? Good question.
Yeah, great question. And we have some internal data on that as well. So if you're interested, you could always send an email to the webinars at matttek.com and we can forward that data to you. But yes, you can measure cytokines from the intestine following your dosing exposure.
Okay, and is there, another good question, is there a matrix where cells are embedded, I'm assuming in the tissue production process?
Yes, so this model is a direct seed model, which is great. So there we use the inserts that can be used in either a hanging or standing platform. And we have a collagen that's deposited to allow for the fibroblasts and epithelial cells to grow.
Right. Have you ever made inflammation studies or performed inflammation studies with this model?
Yes, so again, we have some internal data for inflammatory markers. If you want to reach out to the email that's shown on the screen, we'd be happy to share that data directly with you and then go into more details about how to do those studies with this model.
Another good question on the endpoints. Do you offer additional endpoints like H&E stain, microscopy, gene expression, etc., for use in toxicological assessment of drug effects?
We have an internal group that can actually do those endpoints such as H&E, as well as microscopy and gene expression. That's part of our studies group. And all of the images that you saw for the characterization were done in-house for that. And that's a common endpoint that a lot of people use to assess the health of the tissue.
Okay, another good question. Would you ever recommend starting with the SMI and then moving to the full thickness after initial slash optimization studies?
That's an interesting question. So I think the only reason to necessarily move after initiation or optimization would be if you found out that maybe you required the fibroblasts to be more in vivo relevant. If you've done all your optimization in the SMI, I think you would want to re-optimize in the full thickness model before making that change.
A great question and a trending topic in biotechnology right now. Do these models come with microbiome?
These models do not currently come with the microbiome. That is an area of interest for MatTek, obviously also an area of interest for a lot of researchers. So continued work is being done in that field to understand how to introduce kind of a microbiome for these products.
Okay, and then more questions about the structure of the model. What type of collagen do we use, and have we ever had M cells present in the models?
Yep, great question. I will have to get back to you on the M cell presence. I believe that we're using type 1 collagen, but we can check on that and respond to you as well.
Okay, and then we're getting some more specific questions. Are the fibroblast cells donor matched to the intestinal cells?
Great question. I do not believe that right now our current donors are matched, but that doesn't mean that we don't have the ability to do that. So that would be a custom product. So if you're interested in matched epithelial and fibroblasts, that's something that our technical team would be able to help you with and see if that's possible for this model.
Okay, and we have a specific question that may require additional consult off screen. The question is regarding inflammation studies, can you suggest an ideal TNFA concentration and time of exposure to see a significant variation in tier values?
Yes, I don't have that memorized. TNF alpha, I don't have the concentration memorized, but that's definitely something we can send you some research and data on that we've done internally.
Okay, and is the model anaerobic or anaerobic in any regions, considering for adding gut anaerobes to the model?
Interesting question. So I don't have the answer for if it's anaerobic in any regions. That's not something I'm aware that we've studied, so we'd have to look into that a bit.
Okay, and do you think that having a dynamic continuous recirculating perfusion of the apical / basal layers could be ameliorative for the tissue differentiation / mimicking of the in vivo conditions?
Yeah, great question. So that's kind of on the organ on a chip side of things. We don't have that in place right now. All of our studies have been optimized with just the standard media changes, but that's always something that would be interesting for us to be able to answer. And we know that there's a lot of research out there that shows having kind of that recirculating movement can kind of alter the model. But in general, we find that our models are very in vivo relevant, even without that recirculation.
More good questions. Keep submitting questions if you have them. Do you make disease models with cells from patients with diseases, for example, IBD?
Yes, so right now we only offer the healthy donors. To my knowledge, that IBD is a bit challenging to build a model from, but we do have some studies that have been done with other customers that have treated the models with certain compounds to induce an IBD-like disease state. Some of our other models that MatTek makes, we do actually offer from diseased patients.
Are the cells normal, non-cancerous? And I believe, yes, they are.
Yes.
And is the 96-well EpiIntestinal model amenable to tear measurement?
Yes. So we have protocols on how to do this, as well as recommendations on what probes to use. But the 96-well plates that these are made in are amenable to tear.
Okay, and the last question that was submitted, do you offer preclinical intestinal models?
So right now, these models are not accepted by the FDA for preclinical replacement of animals. That's something that is a big push for MatTek, as well as other companies that are trying to move away from animal models. So this model isn't necessarily approved by the FDA, but you can use it in and work with your regulatory officials to understand how you can potentially use it as a replacement or kind of a pre-test before you need to move into animals.
Okay, another new question. Since the tissues are three-dimensional and the membrane is translucent, can you suggest other live techniques to monitor tissue growth and behavior, excluding tear?
Yes, so tear is a great one just because it's non-destructive. If you're okay with potentially, maybe more destructive endpoints, MTT is a good option. You can also use Lucifer yellow as an alternative to tear, which would be non-destructive. And then standardly, measuring cytokines to tell you if the cells are reacting differently. And then for growth, you can probably use a live cell kit. I would have to check to see how frequently that's been done by people, because MTT is our standard for cell health that's destructive.
And back to the preclinical models topic, have we ever compared the preclinical models in a similar fashion as in the Peters et al. Paper?
Yeah, so the data that was shown was conducted between Peters et al. And MatTek, so that data was actually collected at both facilities. Quite a bit of it was collected at MatTek. So that is our data for understanding how these models compare to preclinical results. And again, that publication has a lot more comparisons between preclinical and clinical. And so we'll send that out in a packet for everyone to review if they're interested, and you can get more information on all of those studies.
Yeah, that's an excellent study. And then there was one question that I missed. It had to do with time in culture. How long can you keep EpiIntestinal in culture for?
So we recommend that it can be maintained for 4+ weeks. Some researchers have taken it out longer. 6 weeks is around the time frame that the Peters et al. Paper was using it for. So we do see high viability for these cells over at least a month in culture.
Great. I think we were able to get to every question. And if no one else has another question to submit, I think we can close the presentation, and then we'll follow up with everybody with the TR packet or the technical reference packet of all of the papers that were cited in the presentation. And if you had a specific question that required us to look back for extra data, especially those who asked about inflammation specifically, we'll reach out to you directly with more information. And as always, you can send an email to the address on the screen, and we'll be happy to chat with you. So thank you so much, everybody, for attending today. Thank you so much, Katie, for an excellent and informative presentation, and hope you all have a good day. Thank you so much.